Hello, this is Anthony Alanis, and I'm going to be presenting my BioProject on coronaviruses and the human angiotensin-converting enzyme 2 receptor. So, there are three main questions I am trying to answer in this study. The first one is, how does the spike glycoprotein of different coronaviruses bind to the ACE2 receptor? And how do the binding complexes differ structurally? And so you can already tell on this slide, we're going to look at how that question could be answered in figure 1. And then we also try to answer the second question on this slide. How do the spike glycoproteins compare to SARS-CoV-2 or COVID-19 structurally? So, and then the last question I'll just go ahead and mention, how are the different coronavirus strains in different organisms or hosts related to each other phylogenetically? And we'll answer that on the next slide. But we'll go ahead and we'll start here. And I'm going to zoom in a little bit so that we can see everything clearly. But so we're going to look at figure 1 first. Figure 1 is it shows 4 different coronavirus spike glycoproteins binding to the ACE2 receptor. And okay, so panel A shows in tan, where my mouse is right now, I hope you can see that. In tan, in panel A, that is the spike glycoprotein to SARS-CoV-1 from 2003. And it is binding to the ACE2 receptor in blue. And then when you look at panel B shows the spike glycoprotein from COVID-19. And this is obviously in 2019 in December, and I think this is the one from 2019 in December, this exact glycoprotein. And notice how similar they are. And we'll see that phylogenetically they're very similar too in their amino acid sequence. And then panel C is it shows the bovine spike glycoprotein binding to the ACE2 receptor. And then panel D shows I think it's civet. Yeah, the civet SARS coronavirus spike glycoprotein. Notice they bind in a similar fashion to the SARS-CoV-1 and 2, but they're just slightly different, especially panel D. It shows a little bit different in civets. The way I did this was it's not the traditional way I think that most people would do this. Most people would use a supercomputer to do binding simulations. I did try to do that using PyDoc, which is like a online server that you can upload a protein database file, and two protein database files, and they'll do the protein binding simulation somewhere in the world that has a supercomputer. They'll literally do it, and then they'll share the results with you afterward. And it's completely free, PyDoc. It's really cool, but I kept running into issues with it because the protein binding that they kept doing, it was really difficult for me to bind the spike glycoprotein to the ACE2 receptor without without making it bind like to this part, the bottom part. It's like, and this is the transmembrane part. It's the part that goes into the membrane, but I don't know how to get rid of the bottom part of these proteins when I'm trying to do the protein simulation using PyDoc. Anyways, so the way I did it was I used the matchmaker command on X Chimera, and it basically superimposes the spike glycoprotein onto the spike glycoprotein from SARS-CoV-2. So I used 2 protein database files. I used one with the ACE2 receptor already bound to the SARS-CoV-2 spike glycoprotein, and then the second protein database file contained whichever coronavirus spike glycoprotein I wanted to look at. So for the first one, I superimposed the SARS-CoV-1 onto the SARS-CoV-2 spike glycoprotein, and then I removed the SARS-CoV-2 receptor or spike glycoprotein from the visualization for clarity, really. And then I did the same thing for the rest of these. So I basically used the matchmaker command, and I think it produced pretty good results. Um, yeah. So, but I ran into some issues, sort of, and I noticed that I could not. So each of these spike glycoproteins is in the open conformation, which is necessary for it to bind to the ACE2 receptor. However, the spike glycoproteins that exist in bats, cats, and birds, I could not find protein database files where they were in the open conformation, which is necessary to show them binding. So I did something else. I instead superimposed this panel A shows in tan the spike glycoprotein from bats, the Rhinolophus bat, HKU4 strain. In tan, that is the spike glycoprotein from bats, and I superimposed it onto the SARS-CoV-2 spike glycoprotein. So you can see they're actually highly similar. You can tell that it has this triad-looking structure on the base. This is the part that makes contact with the ACE2 receptor binding domain. And then I basically rotated it about a horizontal axis. You can tell with this little arrow how I did it. And that's what it looks like from the side. And then I did the same technique with each of these. So panel B was a cat. Notice that these little corners of the spike protein go outwards a little more. However, it does retain a triangular structure on the base that makes contact with the ACE2 receptor. And then when you rotate it, it's about the same height, same width, everything like that. And then the panel C is the avian coronavirus. And it does have a triangular structure. However, most of the most of the residues, the protein residues, amino acids that align with that of SARS-CoV-2 shift these corners. So right here, this is probably where, right in the middle is where most of the overlapping happens between the two spike proteins, or the area of most similarity. And what that does to the model is it shifts these corners a little bit, so it's a little strange. But it does retain the same size and width, same height and width. So they are pretty dang similar. They're all pretty similar. And so I like this experiment a little bit because I got to see, how similar COVID-19 was to other coronaviruses. And yeah, so I hope that does answer. For me, it did answer mostly questions 1 and 2. How do they compare structurally? Their binding complexes, and also just the structure of the spike glycoprotein itself. Now, the third question, how are different coronavirus strains in different hosts or organisms related to each other phylogenetically? So this this third slide is showing the phylogenetic tree of of every single spike protein that I showed in the previous slide, and then some. There's actually more. I use 8 different strains of coronaviruses, and I have off to the right this table showing which ones they refer to in the figure off to the left. So group 1 is SARS-related coronavirus in civets, and then we have SARS-CoV-2 in humans, SARS-CoV-1 in humans, feline coronavirus in cats, which we saw on the previous slide. We also saw civet on the previous slide. The canine coronavirus or alpha coronavirus 1 in dogs, beta coronavirus in cattle, and yeah, so there you go. And so here we see the phylogenetic tree. I made the phylogenetic tree using the, I think it was the quasi-species pipeline on the pipeline thing. And what I did was I basically did the birth-death speciation process, and I did it for amino acids. I took the protein database amino acid sequence for every coronavirus. I took 4 files per host at different points in time as well. So I noticed that, like right here, see how these branches differ in how far they go off to the right. Here they're pretty close to each other because SARS-CoV-2 is a relatively new virus, so there's not a lot of space between these branches. However, when you go down here, notice that with these green nodes, they're all sort of separated horizontally because I took samples from different points in time so that I can see the relationship better phylogenetically. And so anyways, from this graph, you can already tell that SARS-CoV-2 and SARS-CoV-1 are the most similar to each other in terms of amino acids than any other coronavirus. However, the most intriguing thing about this tree is that you can see that the SARS-CoV-1 and SARS-CoV-2 coronavirus strains are more similar to every single one off to the right here. And so what I'm saying is that these right here are more similar to birds, cattle, dogs, and cats, which are in close contact with humans, than they are to bats and civets, because they share a more common ancestor, or a more recent common ancestor than it does with these two organisms. So off to the left, we have civets and a bat, and these are the hosts for these strains of coronavirus. And so, but they share more distant common ancestors to COVID-19, which we're dealing with today. However, COVID-19 shares a more recent common ancestor right here at this node with all of these hosts, so cattle, birds, dogs, and cats. So it poses the question of, okay, well, maybe there was some sort of spillover event at one point in time. Maybe it was with SARS-CoV-1 back in 2003, where we infected birds or cattle or dogs and it spread to each other that way. So I thought it was important to bring up that tree. And so let's see, down here I mentioned that SARS-CoV-1 and SARS-CoV-2 bind to the ACE2 receptor in a similar fashion on the previous slide, panel A and B. And since they do bind similarly to the ACE2 receptor, it raises the question of, well, if they are similar in structure and binding complex, like they bind to the ACE2 receptor in a very similar fashion, and they also are close to each other on this phylogenetic tree, therefore it raises the question, based off of this tree, whichever coronaviruses are closer in relation to these might actually be more similar in binding to the ACE2 receptor than, say, these, the civet and the bat hosts. So finally, the conclusions of our project is I try to tie together the phylogenetic tree and what we saw with the protein binding models or complexes. So the phylogenetic tree indicated that SARS-CoV-1 and 2 are closely related to each other, and that they are more closely related to each other than with any other coronavirus considered in this study. And also, we already mentioned this actually, that in the protein binding model we had on this slide right here, we saw panel A and B that SARS-CoV-1 and SARS-CoV-2 bind to the ACE2 receptor in a very similar fashion, and whereas the structures of the feline infectious peritonitis virus, or basically the cat coronavirus, spike protein and the avian spike protein differed greatly. So I think this is the avian one in panel C, and then this is the feline coronavirus in B. They're very different in structure to A and B. Yeah, so that's all I really wanted to say there, because you can tell visually on those two figures that they're very different. And then lastly, the bovine spike glycoprotein appears to bind to the ACE2 receptor in a similar fashion to SARS-CoV-1 and 2. In contrast, though, the civet spike glycoprotein bound at a much different angle than the SARS-CoV-1 and 2 and bovine coronavirus. So that statement refers to figure 1. I'm basically summarizing what I already said earlier, that these two bind in a similar fashion to A and B. However, C looks more similar to A and B than D does. And basically, I'm saying that the bovine coronavirus, which is C, looks more similar to A and B than does the civet coronavirus, which is panel D. So that was that last conclusion we made in this slide. And then lastly, these are all the accession numbers for the protein database files I used. Actually, no, they're not protein database files. They're from the NCBI database. So yeah, there you go. Thank you so much, and I hope you have a great day.